Now, coming to the Ctl mechanism. If we look into the Ctl mechanism, so this is a target cell, and these are different cytotoxic T lymphocytes. And you can see that there are 4 categories of engagement basically occurring. One is the granule exocytosis pathway, by which a cytotoxic T lymphocyte can kill a target cell. The other is the fast pathway, by which a cytotoxic T lymphocyte can kill the target cell. And the third pathway is the production of cytotoxic cytokines such as TNF or lymphotoxin. So it's a production of TNF or lymphotoxin. So when and these are the 3 basic pathways in which a cytotoxic T lymphocyte can provide death to a target cell. Now, coming to the granule exocytosis pathway. In the granule exocytosis pathway, as you can see, this pathway refers to the release of the contents of the cytotoxic T lymphocyte, cytotoxic granules. Okay. So among these granules are perforin and granzyme. Now, perforin is a pore-forming protein, and granzymes a family of serine protease. It is, however, not clear as to how these molecules enter the membrane, but what has been studied is that once they are delivered to the cytoplasm of the target cell, they are found in this kind of endocytic endosomes. So they are most probably taken in via the target cell via endosomes. And once they reach the cytoplasm, the perforin then facilitates the release of the granzyme molecules from the endosome. There are 2 categories of granzymes that has been observed till date. Granzyme A initiates caspase-independent pathway of DNA damage, while granzyme B triggers classical caspase-mediated apoptosis. So this is basically the granule exocytosis pathway. Now coming to the FAST pathway. We all know that FAST is a transmembrane death receptor that is widely expressed on mammalian cells. Engagement of the FAST on the target cell to the FAST ligand expressed on the armed cytotoxic T lymphocytes result in the death of the target cell. You have studied apoptosis, so the death is done. It's mediated through the death receptor pathway. There is another death receptor pathway operative, that is when the cytotoxic T lymphocytes produce 2 very vital categories of cytokines, which are known as cytotoxic cytokines, specifically TNF alpha and lymphotoxin. Both TNF alpha and lymphotoxin can bind with the tumor necrosis factor receptor 1. The tumor necrosis factor receptor 1 also opts or signals via the death receptor pathway, bringing in about apoptosis. So the main mode of killing the target cell by an armed cytotoxic T lymphocyte is apoptosis.
Now, active target cell is
now we are I'll be taking a particular point of antigenic shift, which has become very vital given the COVID scenario we are going through. This is a classical case of influenza. Now what happens? A common way of a virus to hide from the host immune system is to change its antigenic stripe over successive generations, expressing antigenically new forms of viral proteins that may not be recognized by an individual's existing memory lymphocytes or antibodies. This rapid modification of viral antigens through random mutation is known as antigenic drift. Now, let us take the case of the example of the virus influenza. What happens in case of influenza virus is that the virus cannot proofread its RNA genome during replication and thus sustain a high rate of mutation. The two important proteins, heme agglutinin H protein and neuraminidase N protein, are the only two viral proteins present on the surface of the influenza virion and are thus subtly different from viral generation to generation. So if we look into this picture, we have the N protein and the H protein. And these are the only two recognizable proteins considered to be taken to be as antigens by our immune system. Now let us look into this hypothetical picture. Now suppose this is one influenza virus with subtype A, as you can see, expressing the N1 variant protein and the H3 variant protein. This is the second category of influenza virus, as you can see, subtype B, expressing the N1 variant of the hemagglutinin and the N2 variant of the neuraminidase. Now, these two viruses are co-infecting the same host cell. So, influenza virus, if you look into the genome of the influenza virus, it consists of 8 RNA molecules. Okay, so there are 8 RNA molecules. So when it is infecting a host cell, the host cell now has a 16 RNA molecules, and there can be varying degree of recombinations between the H1, H3 and N2 and N1. So that the new subtype, as you can see, if you look into the outer coat of subtype 8, it exhibited the pink variants and the blue variants. Now you look into the new subtypes, due to the recombination extensive recombination occurring between those molecules, we are having a variety of subtypes. So this is subtype C, which is a combination of both pink and blue, and subtype D, which is again a combination of pink and blue. Now look at the epitopal structures they have shown. We are getting pink blocks and half semicircle pink blocks. We are getting blue semicircle blocks and blue rectangle blocks. Now, when we look into this subtype, we are getting blue rectangle blocks and pink semicircle blocks. And in the subtype D, pink rectangle box and blue semicircular blocks. So, by a simple mechanism of recombination, two subtypes in one cell is giving rise to another variant type of subtypes. Now, when the infection had occurred in the previous case, when it is a first case infection, antibodies were, produced against the pink rectangles, pink semicircles, blue rectangles, blue semicircles. Now, the situation has changed. So, these neutralizing antibodies that were present in the body as memory cell, as a result of the first infection, are becoming redundant, useless, when the fresh subtypes are now released as progeny viruses. So this antigenic drift, or as a result of the antigenic drift, the virus has undergone an antigenic shift. So the shift of the antigen has resulted in the incapability of the previous antibodies to recognize the fresh antigens, and thus, even though there were vaccines produced for subtype A and subtype B, the vaccines becomes useless for subtype C and subtype D. Now, this kind of antigenic shift occurs very rapidly in case of at least influenza virus.